Welcome back to BioMindscape. Here we are going to solve the Re-NEET 2026 questions. So here, how many theca are present in each lobe of a typical bilobe angiosperm anther? We have to tell the answer. Here option A is 2, option B is 6, option C is 8 and option D is 12. Answer the question. So here the correct answer is 2. A male reproductive organ of flowering plants include the anther, and a typical angiosperm anther is bilobe, having two main lobes, and each lobe of anther that consists of an internal chamber we call it theca. Then next question is, the muscle contraction is initiated by a signal sent by the central nervous system and by the release of which of the following? Option A acetylcholine, option B acetyl coenzyme A, option C cyclic guanine monophosphate, and option D is cyclin adenine monophosphate. So here the correct answer is acetylcholine. Acetylcholine is option A is correct. Which one of the following statement about Lac-Operon is correct? Here option A, gene I is consecutively expressed. So this is correct option here. A option we directly get the answer. In Lac-Operon, in transcription regulation system in E. coli that involve in lactose catabolism. So here it contains a structural gene like X, Y and A, and that is a promoter, that is an operator, and a regulatory gene is present. So here the lactose or L-lactose that act as an inducer. It binds to the repressor protein, and after that what happens? After binding and inactivating it, it prevents it from binding to the operator. Thus here B option is incorrect and D is also incorrect. Why? Because galactose, that is the product of the lactose hydrolysis and cannot act as an inducer. Then C option, the I gene, that is regressor gene. It has its own independent promoter and it is transcribed separately. So the structural gene X, Y and Z, Y and A, they share a separate single promoter. And they do not share one single common promoter with the I gene. So your C statement is also incorrect. Which one of the following is female gametophyte of an angiosperm helping in guiding the pollen tube for fertilizing the egg? So here the synergids, it is the female gametophyte of an angiosperm that helps in guiding the pollen tube for fertilizing the egg. So option A, option C and option D are incorrect here. Which one of the following plant produces non-albuminous seed? Option A wheat, option B maize, option C barley and option D pea. So here pea is the particular plant that produces non-albuminous seeds. If the diploid chromosome, next question is, if the diploid chromosome number of the typical endosperm is 36, what would be the chromosome number of its endosperm? Now we have to calculate that to determine a haploid chromosome number. The diploid chromosome number 2n of a given angiosperm is 36. We know that, and we have to find the haploid chromosome number. So what we will do, the chromosome number n divide by the diploid number. So 36 divided by 2, that comes out to be 18. Then what will be the next step? Here during the double fertilization, one haploid male gamete fuses with the diploid secondary nuclei that form the true polar nuclei. We know that. So first this process we call it triple fusion, so which form the primary endosperm nucleus, that is pen. So the ploidy, the endosperm is diploidy, 3n. So what we will do, 3 into 18, that is n. 3 into n, that is 3 into 18, that comes out to be 54. So here which option shows 54? Here we get the answer, option C, that is 54. Which one of the following statement about the reabsorption process in the Henle's loop are correct? We have to tell. So here option A is the descending limb of Henle's loop is permeable to water but almost impermeable to electrolytes. Here, is this option correct? Yes. Then urine get concentrated in Henle's loop. This is also correct. Absorption of sodium and water takes place in the Henle's loop. Question is, which one of the following is not an evidence for evolution? So here the answer for this question is, first is convergent evolution of trait like wings of the bats and butterfly. Option B is paleontological evidence from the fossil record. Option C is embryological support for evolution as proposed by Ernest Haeckel, and option D is divergent evolution of anatomical structures such as the forelimb. So here the correct answer is C, that is embryological support for evolution, that was proposed by Ernest Haeckel, and it is not an evidence of an evolution, whereas all the other options, they are the evidence of evolution. Now moving on to the next question. Given below are the two statements and we have to tell which one is correct. So here the first statement is, the modern Homo sapien arose in Africa and moved across the continent, and option 2 is Homo sapien arose around 75,000 to 10,000 years ago. So if we see, both the statements are correct. So we will go with option A here. Both the statements are correct. Now moving on to the next question. Consider a population of 10 million cells and given the per capita birth rate, that is 0.002 unit times, and the per capita death rate that is given, that is 0.002 per unit time. So the expected number of cell after the 10 generation is, so your option A is 1 million, option B is 5 million, option C is 10 million, and option D is 100 millions. So here the correct answer is 10 million. How we got this answer? dn by dt is equal to rn. So r is equal to b minus d. The parameter r is the intrinsic rate of the natural increase. So we have to calculate it. R is equal to b minus d. So here b is what? The per capita birth rate, and d is the per capita death rate. So birth rate minus the death rate, we will get birth rate, it is given in the here. The per capita birth rate is 0.002 and death rate is also 0.002. So that comes out to be r is equal to 0. So to determine the r is equal to 0, that is population growth when the intrinsic rate increases, r is exactly 0, the rate of the change of population, that is dn by dt, that is 0 into n, which comes out to be 0. So we have to calculate the population after 10 generation, that is nt is equal to n0. Here the initial population n0, it is 10 million. The expected number of the cell remains, that is 10 million, which is the option C. Now next question is, during PCR the primer binds to the DNA strand in that step. When does the primer bind? Option A, denaturation, option B, extension, option C, annihilation. So here annealing is the step where the primer that will be binding to the DNA strand. Here we are going to find out the length of the stem at a given time. So 0 is 20 centimeter and the arithmetic growth rate, that is given 30 centimeter per day. What is the length of the stem at the end of the 7 day, we have to tell. So here in this question we have 4 options and we have to tell which one is correct. So let's solve this question. First, length that is equal to L0 plus RT. So here initial length that comes out to be 20 centimeter, the growth rate that is 30 centimeter, and the duration, that is time, is 7 days. So 0 to 20 centimeter, arithmetic growth rate is 30, that is r is 30, time is given 7 days. Okay, so the formula for this is length is equal to 20, that is the initial length, plus in bracket 30, that is the growth rate, into the 7, that is the days that are required, so 7 days. So 20 plus 30 into 7. So what do we get? L7, that is 20 plus 30 into 7, 210, that comes out to be 230 centimeter. So here the answer, the length of the stem at the end of the 7 day is 230 centimeter. So here we can see which option is showing 230 centimeter, that is option C. So the length of the stem at the time 0 is 20 centimeter and the arithmetic growth rate, that is 30 centimeter per day, and we have to tell the length of the stem at the end of the 7th day, that is 230 centimeters. So we get the answer 230 centimeters. C option is correct. Synopsida class belong to which one of the following? That is the pteridophyte. It belongs to the pteridophyte. They are the vascular cryptogam, non-seed bearing plants that contain the xylem and phloem, and they are classified into 4 distinct classes based on the evolution and structural characteristics. So here the Synopsida, that is Coleoterm, then Lycopsida, that is Selaginella and Lycopodium, and Synopsida, that is Equisedum, and Pteropsida, that is the Pteris and Adiantum. Which one of the following statement regarding the photorespiration are correct? We have to tell. So here, do not occur in C3 plants. This is wrong. Photorespiration occurs specifically in C3 plants under high temperature and lower CO2 condition. It is absent in C4 plant, thus A is incorrect. Now let's see the second option. CO2 is consumed and O2 is generated. This is true. Photoglycolate is formed. This is true. No synthesis of ATP and ADP. So here B, C and D, these are the correct option, and others are A option is incorrect. Next is smooth ER. What is it? It is a major site of synthesis of the lipid. So here B option is correct. It is a major, smooth ER is the major site of synthesis of lipid. Here A and C option we can see. What are they? Has a ribosome attached to its surface and it is actively involved in protein synthesis. So A and C, they are describing the rough ER, not smooth ER. And D option, it describes the Golgi apparatus or the plastid, where it is a site of synthesis of carbohydrate. Which is the site of synthesis of carbohydrate? Golgi. And here the has a ribosome attached to its surface is the rough endoplasmic reticulum, and it is actively involved in the protein synthesis. Which one of the following statement is incorrect? We have to tell. Here alpha cell of pancreas secrete the glycagon, then alpha cell of pancreas secrete insulin. This is wrong. The second option B is incorrect here. Why? Because alpha cell of secrete insulin is secreted by the beta cell, not alpha cell, and the glucagon is secreted by the alpha cell. So statement B is incorrect. Other statement are correct here. Glucagon stimulate the glycogenolysis and B cell of the pancreas secrete the insulin. Next is the genus that represent a group of closely related species. Group of closely related species is a genus. A population of plant and animal, what is it? It is ecological community, an individual plant or animal, that is single organism, and group of closely related family is order, not genus. Which one of the following is not prokaryote? Bacteria, blue-green algae, mycoplasm and fungi. So here the fungi, that is not a prokaryote, while the bacteria, blue-green algae and mycoplasm, all those are the prokaryotes. Fungi is diverse kingdom of multicellular, unicellular organism, eukaryotic, heterotrophs. So these cells possess different nuclei, mitochondria and chitinous cell wall. Which one of the following plant growth regulator promotes the internode elongation prior to flowering in the cabbage? That is the gibberellin, that follow the plant growth regulator that promote the internodal elongation prior to the flowering in the cabbage. AB is a growth inhibitor. Indole butyric acid is in auxin and it is involved in the root initiation, whereas ethophone it is an ethylene releasing agent used for the fruit ripening. So gibberellin is the correct hormone here. The corresponding sequence of adult cell cycle is G1S, G2 and M5. Then we have to match the pairs here. The fusion of protoplasm between the gametes, that is the plasmogamy, then fusion of two nuclei, that is the karyogamy, and generation of the haploid spore, that is meiosis. Then here two statement. The class of the name of reptile refers to the creeping of crawling mode of locomotion. This is correct. And option 2. All organisms belong to the reptile have 3-chambered heart. This is incorrect. Does reptile have a 3-chambered heart? All organisms belonging to the reptile have a 3-chambered heart, but here we can, if we evaluate this statement, the reptile typically have 3-chambered heart, 2 artery and 1 partially divided ventricle. However, crocodile, alligator and gharials possess 4-chambered heart. Because of these major exceptions, all reptile have a 3-chambered heart. This statement is incorrect. Then given below are two statements. Chromosomes are fully condensed at the end of the prophase 1. This is correct. And meiosis 2 resembles mitosis. This is wrong here. The meiosis 2, it is an equational division where sister chromatids separate, which is functionally equivalent to mitosis. In contrast, meiosis 1 is reduction division, that is involving homologous chromosome segregation and crossing over, making it fundamentally different from the mitosis. So here meiosis 1 does not resemble mitosis. So the second statement is incorrect. Which one of the following is not a characteristic of the chordates? Absence of the gills is not a characteristic of the chordates here. Whereas presence of notochord, central nervous system in dorsal, and presence of post and tail, this is correct option here. So C is the correct option. It is not a characteristic of the chordates. Welcome back to BioMindscape. Here the question is, if a population with n, that is 400 individual, it increases in number till it reaches an k, that is 500 individual. So k being the carrying capacity, assume the intrinsic rate of nature increases to be 0.01. What would be the population growth rate? Now here we have to do some calculations. So let's do it. So first dn by dt, that comes out to be Rn k minus 1 n upon k. So here R is equal to 0.01, n is equal to 400 individual, and the k, that is 500. So we will put it in the formula. Dn by dt, that comes out to be 0.01 into 400 in bracket 500 minus 400 upon 500. So it comes out to be 4 in bracket 100 upon 500. So final answer that we get 4.02. Here we get pointed. So pointed is the answer. So let's check the answer. So the option A, that is, so we got the answer here. First question is, which type of polymerase activity is shown by the enzyme telomerase? So the activity that is shown by the enzyme telomerase is RNA dependent DNA polymerase. First, what is a telomerase? It is a specialized enzyme that is responsible for maintaining the length of the telomere, which are repetitive nucleotide sequence at the end of the linear chromosomes. Chromosome, they are short with each nuclear division, and telomerase counteract this by adding the telomere repeats to the chromosome end. So how do we know that RNA dependent DNA polymerase is the correct answer? So here the DNA dependent DNA polymerase, it occurs when an enzyme that synthesizes the DNA using a DNA template, example DNA polymerase during replication, and DNA dependent RNA polymerase, it occurs when an enzyme that synthesizes RNA using a DNA template, example RNA polymerase during the transcription. Whereas RNA dependent DNA polymerase, here an enzyme that synthesizes the DNA using an RNA template. So it correctly describes the telomerase. Whereas the RNA dependent RNA polymerase, here it defines an enzyme that synthesizes the RNA using an RNA template that is found in some viruses. Now let's see what we know the answer is option C here. The activity of telomerase, it is synthesized by the DNA using an RNA template which is defined as RNA dependent DNA polymerase activity. Moving on to the next question. So next question is, purine found in both DNA and RNA are option A cytosine and thymine, option B adenine and thymine, option C adenine and guanine, and option D guanine and cytosine. So here the correct answer is adenine and guanine. These are the purine form in both DNA and RNA. So what is different? The pyrimidine. In RNA it is uracil, whereas in DNA it is thymine. So we write it T in DNA and U in RNA. Then next question is, if the distance between the two consecutive base pairs is 0.04 nanometer and total number of base pair of DNA double helix is typically mammalian cell is 6.6 into 10 raised to 9 base pair, then the length of the DNA, we have to find out the length of the DNA. For that we will calculate this. So we'll write the length, that is 6.6 into 10 raised to 9 base pairs into the 0.04 will be converted into this nanometer. We will be converting it into 10 raised to 9 here base pairs, that is 0.34 into 10 raised to 9 base pairs minus 9 base pairs, which comes out to be 2.24 meter. So let's see which option shows 2.2 meter. That is option C here. So C option is correct here and we get the answer 2.2 meter. Next question is DNA fragment. What are they? Positively charged, negatively charged, neutral, or depends on the size? The fragment of the DNA, what are they? Negatively charged. Then in a DNA strand, the nucleotide, they are linked together by which type of bond? Glycosidic, phosphodiester, peptide and hydrogen. Which types of bond are there? The DNA strand in the nucleotide, they are linked together by the phosphodiester bonds.